One. All right. Today we are talking about bye. Godspeed. Strength and honor. Good luck. We miss you already. Why are you murdering me with your eyes?
I'm literally gonna fight him. That's a time one more time. Please grab his.
You can vote her off the island.
I will. I'll tell your coach you're not going anymore. Tell her you quit. Okay.
Alright. So the thing about cell membranes, all cells have them. No matter what type of cell it is. What about prokaryotes, though? Yes, they do. What about fungi? Heck yes, they do. But what about plant cells? Pero Senor Dreads, a plant cell has a cell wall. It also has a cell membrane. Okay? Plant cells have them both. Because if a plant cell didn't have that cell wall, it would rupture when it fills up with water. Okay? So that's why a plant cell has both. So all cells, regardless of their type, have this cell membrane. And you remember what's on the inside? Those little compartmentalized structures on the inside, what they're called? Starts with an O, ends with organelles. Organelles! Okay? Most organelles are made of membranes. Or they at least have them on the outside. There's two special ones. The mitochondria and the chloroplasts have two. They're like the ultimate hoarder. I'm going to draw a mitochondria for you. Why? Because I can. So this mitochondria has two membranes. It's got an outer membrane and it's got an inner membrane. Do they look the same? Nah, what's different about them? They're the same depth, so they're the same thickness because they're both made of a phospholipid bilayer. But just kind of looking at it, what's different about them structurally? The inner one's a lot more wrinkly. Okay? So this inner one, so I'm going to draw an arrow. I'm going to say this inner one is highly folded. So it has a lot of folds on it. Let's say, as I'm looking at this, on the membrane, where the inner membrane is, that's where I make ATP. So that's the location of ATP synthesis, is on that inner membrane. Talk with your partner or a group or yourself if you hate your partners. Why do you think the mitochondria has evolved to have a highly folded inner membrane if that's the location where ATP is made at? That's the location where ATP is made at. So why do you think, over millions and billions of years, why do you think this mitochondria has evolved to have a very highly folded inner membrane, which is the location where ATP is made at? Why do you think that's advantageous to making ATP?
We need to confuse That's a beautiful answer. It's rejected.
Oh.
Is it confused viruses at one side? If a virus where was it one side? No, maybe it's no, never mind. I don't know. It's projected. It's cute.
Alright.
It does. Look.
Let's say this is my outer membrane of my mitochondria. Okay, so you see it's not really highly folded, so let's say it's like this. Let's say this is my inner membrane. If that inner membrane wasn't folded, alright, it would probably go in here like this. Okay? And it would fit in there like that. Let's say every inch of this membrane, just for the sake of this, every inch of this string can make 1 ATP. This is roughly 9 inches long, so how many ATP can I make? 9. Well, let's say that I've got this one. And let's say I fold it now. Okay? So I take this membrane, or this string, and I fold it and fold it and fold it and fold it and fold it. And so I highly fold this thing over and over and over, and you get the point. And it goes on the inside very highly folded. But then I take this thing, and now I've got this 6-foot cable that's 72 inches long. Now how many ATP can I make? 72. So if I increase the amount of folding, the more folded something is, what I increase is I increase the surface area. And if I increase the surface area of something, I increase its efficiency. So if I have in a highly folded inner membrane, it increases the surface area, which increases the efficiency, meaning I can make a heck of a lot more ATP in the same size organelle than I could if it wasn't highly folded. So highly folded equals more surface area, which leads to more ATP. ATP is the energy molecule. That's what our body uses. It's called adenosine. Ooh. I just drank Pepsi. That was a burp and like a hiccup at the same time. Hurt. Yeah. More ATP. ATP is an energy molecule. It's called adenosine triphosphate. That's how our body gets our energy, is this molecule called ATP. Okay? Questions? Okay. Let's talk about our next thing, the model of this. Before we draw the model of what ATP looks like, we should probably name it. It's what's called a fluid mosaic model. Anyone heard that before? Okay. Fluid mosaic model. What does fluid mean? So fluid means it's like movable, or not rigid or stiff. A cell wall is very rigid or stiff. That's why like bark on a tree or a tree trunk is very stiff. It's very dense amounts of cell wall. A cell membrane is not, though. A cell membrane is more like a water balloon. It can move around. Thus the fluid-like. Where then our next word, you may be familiar with it, you may not, but all of you know what a mosaic is.
Is that like art or something?
Okay, it is something like art. Think throwback to like kindergarten or first grade. When your teacher's like, okay kids, we're going to make a Valentine's Day card for your parents. Get out the macaroni and the feathers and the beads and glitter and all this stuff and glue all that crap together. What mosaic means is made of many different things or many different parts.
Bless you.
Mosaic mate's made of many different parts. So fluid mosaic model means I've got this cell membrane that can move around kind of like the surface of water. And it's made of all these different things. And so now we're going to draw these different things. Ready? Time to see your arts and crafts. Actually, I'm going to draw a phospholipid first. Oh, we already drew phospholipid last unit. When we drew lipids. Okay. So I've got some phospholipids. I've got a protein. I've got some more phospholipids. Right here, I've got this little guy. Called cholesterol. Oh. You and me both, brother.
Do you have green?
Oh my god.
You don't have to use the same colors as me.
It's all green.
Okay. I'm gonna draw one more. Can I scroll up? I don't know if you said scroll up or grow up. Fair enough. What drinks do balloons hate? Pop. What's a dog's favorite pizza? Pepperoni.
One of them.
They already messed with the picture.
How do you find King Arthur in the dark?
Hey.
With a nightlight.
I was gonna say that. I swear.
Why shouldn't pizzas tell jokes? Because they're really cheesy. All right, you ready to label this bad boy? Yeah, we are. Okay. So, okay, we will label not with that color. I do not like that color. Okay, here we go. So, if I'm looking at this thing, I've got this part right here of a phosphate head. That phosphate head is polar, meaning it is, this zone right here is what's called hydrophilic. What about this part right here? It's also hydrophilic. But what about the middle? Hydrophobic. So I've got these areas of charge, hydrophilic, hydrophobic. Okay? And now I'm going to go to this next thing right here, this big green one. This big green one is what's called a transmembrane protein. Transmembrane protein. Have you ever heard of a transcontinental railroad? Yeah, you have. What the heck is it? Oh, that's what I was hoping for. It travels across the continent. So, thus transcontinental. Trans means across. That prefix trans. So what do you notice about this protein? It goes across the entire membrane. It goes across the entire phospholipid bilayer. All right? But about properties of this protein. Do you think it's polar or nonpolar? I got some polar. Anyone think it's nonpolar?
Wait, what are we looking at again?
This green protein, this transmembrane protein.
It's both.
What?
Wait, what is
What about you with this nonsense?
I'm kidding. I'm not gonna know. I don't know.
All right. Any of you hear what Ulysses said?
Hey, nah.
He was right. Are you still kidding?
No, what the heck?
He was kidding.
I was like partially kidding.
He said it's both. Because some people are saying it's polar, some people are saying it's non-polar. It's both. Because right here, it's polar. Why does it have to be polar right there? Because it's right next to those polar heads of those phospholipids, so it's got to pair up with them. In the middle, it's what's called non-polar. Why? Because those lipid tails are non-polar. And then that's polar. I can't remember how to spell my word. I had to write it down earlier. I don't know if it's still over here. I can't even say the stupid word.
Hey, don't say it at all.
Is it? A-M-P-H-I-P-A-T-A. Nope. Yeah, P-A-T-H-I-C. Okay, so I've got a side note. This transmembrane protein, because it's got an area that's polar and an area that's non-polar, so it's got these different areas of charge, this transmembrane protein is what's called, I'm just going to write it right here. I'm going to write trait amphiphaphic. A-M-P-H-I.
What the heck, man?
So it's both hydrophilic and hydrophobic. Think about your, when we built a protein out of our pipe cleaner, proteins were long repeating chains of what? Don't say lipids. Amino acids. And what represented the amino acids? The little beads. Did the beads behave differently? Yeah, they did. And so what happened is, were some of the beads polar? Yeah. Were some of the beads non-polar? Yeah. So what it does is it causes itself to fold a certain way. The same thing happens with this transmembrane protein. Some of the beads are going to fold, or some of the amino acids are going to fold away, so they're on the outside, the polar ends, because there's water on the inside of the cell and there's water on the outside of the cell. All right, so it's going to orient itself so the non-polar amino acids are on the inside. That's what's called amphiphaphic, meaning it's got areas of polar and areas of non-polar. Then I've got this bad boy right here. We always associate it with being bad, but it's actually good in moderation. We have to have it. Cholesterol. Cholesterol. Any clue what it does? Any clue what cholesterol does? What'd you say? Cholesterol maintains membrane fluidity during temperature change. So, maybe because it's right. So, when it gets really cold, stuff tends to condense together. We don't want that cell membrane getting too rigid. It's going to help keep those phospholipids apart so it still has that fluidity. Because if they got too close together, that cell membrane would get too stiff and it wouldn't work right. Also, if it gets too warm, stuff tends to separate out. Cholesterol holds those phospholipids together. So cholesterol is a good thing. We need it to help maintain membrane fluidity. Then we've got this guy right here called a peripheral protein. Any guess what that might mean? Peripheral. Have you even heard the word peripheral?
No.
Yes, you have. What does it mean? It's your side vision. All right? So, it's like if I'm over here, I can see Ulysses and I can see Amia, and I'm looking straight. But I've got peripheral vision that I can see stuff on my side. And so peripheral means side, but why do you think this is called a peripheral protein? It's only on one side. It's not transmembrane like the other one. It's only on one side. Okay? And then finally, I've got this one right here, which I know a lot of you are going to struggle with what it means. It is a protein with what attached to it? Sugar. Because glyco means sugar. Okay? So tomorrow we're going to go over what a lot of these things do. I'm going to write it out for you guys so you can have it nicely laid out. Glycoproteins are used for cell communication and recognition. Okay? But long story short, this is your fluid mosaic model. These are all the big components of it. And in the words of Mr. Anderson, I hope that was helpful.
Bianca, are you going home today?
Click here to